This is the ECRs take-home messages video. So over on the right, I've got an abbreviated pathway which highlights some of the things we've been focused on for our enzyme-coupled receptor example, which is the receptor tyrosine kinase. And so here's kind of the things that I want you to see as a bigger picture about this. It might help you to organize your information and see why we're talking about these things. So first off, phosphorylation and kinases are a theme. You just can't get away from kinases and therefore from phosphorylation through this topic. And that starts off with the receptor tyrosine itself, which has a kinase domain and then performs cross-phosphorylation. This extends to other enzymes, for example, P13K, which is a kinase, and the phosphate groups, which are present on PIP2 and PIP3. Also, AKT is a kinase, and therefore is going to phosphorylate or not BAD. And let's not forget about the MAP kinase cascade, which obviously has kinases. So that's probably the biggest theme for the receptor tyrosine kinases. There's also some other things that we can check out about protein dynamics and how receptor signaling works, and some things you should keep track of. So very importantly, note the difference between an individual protein, which would be a monomer, versus a multi-unit protein, for example, a dimer. Also note that there are certain domains within each protein. Sometimes we'll keep track of those domains, and some of the ones that I can highlight right here, great example is the receptor tyrosine kinase itself. So it's a monomer, as you know, when it's inactive, dimer when it's bound to its ligand, for example, a growth factor, and then within each of the receptor protein monomers, there are several domains, including the kinase domain, which is down here. There are also we also highlighted the adapter proteins and that they might have SH2 and or SH3 domains, and so those would be over at this position here in the pathway. And so make sure that you know exactly where those are. Be really careful. I'm going to be super anal about if you're going to circle domains that you get just that particular portion. Don't circle extra proteins, because circling extra proteins is going outside of the idea of a domain, which is just a subset of one protein. Next, let's talk about the types of amino acid residues, so AA residues. So the types of residues matter, which are found within a particular protein. Obviously, they matter for protein folding and protein function, but in particular for all this phosphate and kinase talk, the important ones to keep track of are the ones that can be phosphorylated. And the only amino acids that can be phosphorylated are serine, threonine, and tyrosine. And again, we've heard a lot about tyrosines due to the fact that the receptor tyrosine kinase itself, that's the amino acid residue that is phosphorylated, but with all these other phosphate interactions going on, including from these different kinases, know that they may be phosphorylating other amino acids along this list. And finally, for this slide, individual residues matter. So not just if it's a tyrosine or not, but exactly which tyrosine it is. I have only drawn one phosphorylated site here, but as you may recall from the textbook, there were, say, five phosphate groups that could be added to 5 different tyrosines. And so each of those particular residues in their exact location makes a difference. We didn't really highlight differences across them as far as any ones that you should know in particular, but just know that the level of detail that is going on with these signaling pathways can distinguish, for example, between the first tyrosine that's in this location versus if there's like a third tyrosine down here. Those would be distinct differences and might affect things like adapter proteins. Continuing on to talk about take-home messages for ECRs, in cell signaling, some proteins have enzymatic roles, for example, the receptor tyrosine kinase up here, or P3K or P13K over here. Those are enzymes, but some other proteins will have non-enzymatic roles, and those include the adapter proteins, which I didn't detail here, or they are also known as scaffold proteins. And so notice that those are 2 different roles, so enzymes are always enzymes. Adapter proteins are always adapter proteins, or enzyme portions of a protein is always an enzyme. And so all of these parts are essential for the receptor signaling to occur properly. Second step here is to talk about how powerful proteins like RAS are powerfully regulated. And in particular, RAS is regulated through 2 different enzymes, and this regulation can turn the enzyme on and then also turn it off to get it to that spot where it's not too active and not too underactive, but just right. And so the names of those Regulatory enzymes are the RAS-GEF and the RAS-GAP. Those are not shown here. Also, I did not draw this properly. Remember that RAS is actually tethered to the membrane. We also see that phospholipids can be an important part of signaling pathways, in particular PIP2 and PIP3 over here. Those are each phospholipids, and so those are necessary as a reactant and a product from this P13K to drive signaling to AKT, and then eventually set forth the messages regarding apoptosis and cell growth. So although we talk about a lot of proteins, occasionally phospholipids also have a key role in the signaling pathway itself as far as perpetuating the message. Also note that growth factors are common ligands for receptor tyrosine kinases, and growth factors, just like their name sounds like, will control cell growth. We've heard about a lot of different types of growth factors, like epidermal growth factor, and so that's abbreviated with a EGF. Also, sometimes you might hear about nerve growth factor, NGF, fibroblast growth factor, FGF. And so just pay attention to this GF and know that growth factors are typically going to correspond to receptor tyrosine kinases. And basically, again, this whole thing is to talk about the cell growth part. And so the way that this ties into cell growth for this growth factor is because there are a couple different ways that the signaling can go down. So one way is that RAS can be activated, and active RAS will tie into the MAP kinase cascade, which eventually cues up mitosis. Remember that the M of MAP kinase stands for mitogen. And so mitogen meaning mitosis generating. Secondly, RTKs can clue into the P13K enzyme, and so then that also ends up determining cell growth versus apoptosis by acting through AKT. And this is a core part of an organism's life, as well as a cell's decision to live or die. And basically, any errors in growth factor, in other words, RTK signaling, can cause a variety of abnormalities, in particular cancer or developmental errors. For example, we talked about the type of dwarfism in which bones do not grow properly due to a error in fibroblast growth factor signaling.